The year was 1983. The world was grappling with a new and terrifying disease-acquired immunodeficiency syndrome, or AIDS. This mysterious illness was spreading rapidly, causing widespread fear and uncertainty. Scientists were in a race against time to understand this mysterious illness, desperately seeking its cause and a cure. Laboratories around the globe were buzzing with activity as researchers worked day and night to unlock the secrets of this deadly virus. Amid this urgency, researchers at the University of Minnesota made a crucial breakthrough: the isolation of a specific strain of the virus that would later be known as human immunodeficiency virus type 1, or HIV-1. This discovery was a beacon of hope in a time of darkness. This strain, designated HIV-1 MN, quickly became a cornerstone of HIV research, offering invaluable insights into the virus's behavior and paving the way for the development of life-saving treatments. The scientific community now had a reliable model to study, which was a significant step forward. The discovery of HIV-1 MN marked a turning point in the fight against AIDS. It was a moment of triumph and relief for the researchers who had been tirelessly working to make sense of the epidemic. Before its isolation, researchers struggled to study the virus, hampered by the lack of a reliable and readily available source. The challenges were immense, and progress was slow. The HIV-1 MN strain, however, could be grown relatively easily in the laboratory, providing scientists with a crucial tool for their investigations. This advancement allowed for more consistent and reproducible results in research. This breakthrough enabled researchers to study the virus's structure, understand its life cycle, and develop diagnostic tests to identify infected individuals. It opened up new avenues for research and potential treatments. The isolation of HIV-1 MN was a collaborative effort, a testament to the power of scientific cooperation in the face of a global health crisis. Researchers from various disciplines came together, pooling their knowledge and resources. Researchers at the University of Minnesota, led by virologist Dr. Robert Gallo, worked tirelessly to isolate the virus from a blood sample taken from an individual with AIDS. Their dedication and perseverance were instrumental in this groundbreaking discovery. Their efforts culminated in the successful culturing of the virus, a pivotal moment that would forever alter the trajectory of HIV research. This achievement was celebrated as a major milestone in the scientific community. The discovery of HIV-1 MN was not just a scientific triumph, it was a beacon of hope for millions around the world affected by the burgeoning AIDS epidemic. It provided a glimmer of optimism in an otherwise bleak landscape. It signified a crucial step forward in understanding and combating this deadly disease. The knowledge gained from studying HIV-1 MN would inform future research and public health strategies. The isolation of this strain provided researchers with a vital tool, a key that would unlock many doors in the fight against HIV/AIDS. It was a foundational discovery that would lead to numerous advancements in the years to come. The HIV-1 MN strain, while belonging to the broader group of HIV-1 viruses, possesses unique characteristics that distinguish it from other strains. These unique traits have made it a focal point of extensive research and study in the scientific community. One of its most notable features is its ability to infect cells in a laboratory setting with relative ease. This ease of infection is not just a minor detail, it has significant implications for how researchers can study the virus. This characteristic, known as high replicative capacity, made HIV-1 MN an ideal candidate for research purposes, allowing scientists to cultivate large quantities of the virus for their studies. The ability to produce large amounts of the virus means that experiments can be conducted more efficiently and with greater consistency. Unlike some other HIV strains, HIV-1 MN can infect a broader range of cells, including cells from different species. This broad cell tropism is particularly useful for cross-species studies and understanding how the virus might behave in different biological contexts. This characteristic proved invaluable in the early days of HIV research, when scientists were still trying to understand the virus's basic biology. The ability to infect various cell types provided a more comprehensive view of the virus's behavior and interactions. The ability to grow HIV-1 MN in different cell types allowed researchers to study the virus's life cycle in detail and develop assays to test the efficacy of potential antiviral drugs. These detailed studies have been crucial in identifying key stages in the virus's replication process that can be targeted by new treatments. Another characteristic that sets HIV-1 MN apart is its relative genetic stability. This stability is not just a trivial feature, it has profound implications for the reliability of research findings. While HIV is notorious for its high mutation rate, HIV-1 MN has demonstrated a comparatively slower rate of genetic change. This slower mutation rate allows for more consistent results in long-term studies, which is essential for developing effective treatments and vaccines. This stability is crucial for research, as it ensures that the virus used in experiments remains consistent over time, allowing for reliable and reproducible results. Consistency in experimental conditions is a cornerstone of scientific research, and HIV-1 MN provides that reliability. This stability also proved beneficial in the development of early diagnostic tests, as the tests could be designed to target specific genetic sequences that were less likely to mutate. Reliable diagnostic tests are essential for early detection and treatment of HIV, and HIV-1 MN has played a key role in this area. The unique characteristics of HIV-1 MN have been instrumental in advancing our understanding of HIV. These advancements have not only deepened our knowledge of the virus, but also paved the way for new therapeutic approaches. Its high replicative capacity, broad cell tropism, and genetic stability have made it an invaluable tool for researchers, paving the way for the development of diagnostic tests, antiviral therapies, and insights into the pathogenesis of HIV infection. The contributions of HIV-1 MN to the field of virology cannot be overstated, as it continues to be a cornerstone in the ongoing battle against HIV/AIDS. In the early 1980s, the world was gripped by fear and uncertainty as the AIDS epidemic unfolded. Scientists were working feverishly to understand this new and deadly disease, and the isolation of HIV-1 MN provided a much-needed breakthrough. The strain's ability to be grown in the laboratory allowed researchers to study the virus in ways that were previously impossible. One of the first and most crucial applications of HIV-1 MN was in the development of diagnostic tests for HIV infection. Before these tests, diagnosing HIV was challenging, relying on clinical symptoms that could be vague and easily mistaken for other illnesses. The availability of HIV-1 MN allowed researchers to develop blood tests that could detect antibodies to the virus, providing a more accurate and reliable way to diagnose infection. HIV-1 MN also played a crucial role in the development of the first antiretroviral drugs. Researchers used the strain to screen thousands of compounds, searching for those that could inhibit the virus's replication. This painstaking work led to the discovery of AZT, the first drug approved by the FDA for the treatment of HIV/AIDS in 1987. Beyond diagnostics and treatment, HIV-1 MN was instrumental in understanding the basic biology of HIV. Researchers used the strain to study the virus's structure, its life cycle, and how it attacks the immune system. This knowledge was fundamental in developing strategies to combat the virus and prevent its transmission. The isolation of HIV-1 MN marked a turning point in the fight against HIV/AIDS. This particular strain of the virus, identified in the late 1980s, provided researchers with a critical tool to better understand the virus's behavior and characteristics. Its unique characteristics made it an invaluable tool for researchers, paving the way for groundbreaking discoveries that transformed the landscape of HIV diagnostics and treatment. The ability to study HIV-1 MN in detail allowed scientists to uncover the virus's mechanisms of infection and replication. The development of accurate and reliable diagnostic tests was arguably one of the most significant contributions of HIV-1 MN. These tests revolutionized the way HIV was detected, moving from a reliance on clinical symptoms to precise laboratory-based diagnostics. Before these tests, diagnosing HIV was a challenge, relying heavily on clinical symptoms that were often nonspecific and could mimic other illnesses. This often led to misdiagnosis and delayed treatment, exacerbating the spread of the virus. The availability of HIV-1 MN allowed for the development of blood tests that could detect antibodies to the virus, providing a more definitive diagnosis and enabling early detection and intervention. Early diagnosis is crucial in managing HIV, as it allows for timely treatment and reduces the risk of transmission. HIV-1 MN also played a pivotal role in the development of the first generation of antiretroviral drugs. These drugs were designed to inhibit the replication of HIV, and their development marked a significant milestone in the treatment of HIV/AIDS. These drugs, designed to inhibit the replication of HIV, were initially tested using HIV-1 MN in laboratory settings. The strain's high replicative capacity and susceptibility to these early drugs allowed researchers to evaluate their efficacy and pave the way for clinical trials. The strain's high replicative capacity and susceptibility to these early drugs allowed researchers to evaluate their efficacy and pave the way for clinical trials. This process was crucial in ensuring that the drugs were safe and effective for use in humans. The development of AZT, the first drug approved for the treatment of HIV/AIDS, was a direct result of research conducted using HIV-1 MN. AZT, or zidovudine, was a groundbreaking drug that offered the first glimmer of hope in the fight against HIV/AIDS. This breakthrough drug, while not a cure, offered a glimmer of hope in the fight against this deadly disease. It demonstrated that HIV could be managed with medication, significantly improving the quality of life for those living with the virus. It marked the beginning of a new era in HIV treatment, one where the disease could be managed, and lives could be prolonged. The success of AZT paved the way for the development of more advanced antiretroviral therapies that are used today. The legacy of HIV-1 MN extends beyond these early breakthroughs. The strain continues to be used in research today, helping scientists develop new and improved diagnostic tools, more effective antiretroviral therapies, and potential vaccines. The strain continues to be used in research today, helping scientists develop new and improved diagnostic tools, more effective antiretroviral therapies, and potential vaccines. The ongoing research ensures that we are continually improving our understanding and management of HIV. Its contribution to the fight against HIV/AIDS is immeasurable, a testament to the power of scientific research and the importance of collaboration in the face of global health challenges. The story of HIV-1 MN is a powerful reminder of how far we have come and the importance of continued research and innovation in the fight against